Today we'll talk about the pleiotropy. What is pleiotropy? This pleiotropy, paleo means many and tropic means effect. So we can say that pleiotropy is simply the multiple effects of a gene. Pleiotropy is multiple effects of a gene. In other words, we can say that the ability of a gene to influence several phenotypic traits is known as pleiotropy. So the ability of what is pleiotropy? The ability of a gene to influence number of traits, number of phenotypic traits is known as pleiotropy. And the gene which has many effects, which has multiple effects, is known as pleiotropic gene. Pleiotropic gene. And the gene which has multiple effects is known as pleiotropic gene. According to the Mendel said that one particular gene, it influences one specific character. But several cases have been found where it was found that one gene can influence several characters, one gene can produce several characters, one gene can influence many phenotypic traits, and the gene which influences several phenotypic traits, several characters, is known as pleiotropic gene. Then we have in case of pleiotropism or pleiotropy, this pleiotropic gene, it can have major effect and secondary effects. It can have major effect and secondary effect. What I mean to say is that sometimes this pleiotropic gene, in some phenotypic traits, it produces more pronounced effect. That is the major effect. And in the same phenotypic trait, in other phenotypic traits, this pleiotropic gene, it produces what is called as less evident effects, what are called as secondary effects. So we can say that a pleiotropic gene can produce major effect in one trait and it can produce less evident effect in the other traits, because a pleiotropic gene influences several traits. In one or two traits there would be a major effect, major influence, or we can say that evident effect, that is called as major effect, and in others there would be little evident effect, that is called as secondary effects. So we have the to discuss this pleiotropism, we will take two examples, or pleiotropy. We will take example of pleiotropy is sickle cell anemia. Sickle cell anemia, we have phenylketonuria, 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 phenylketonuria. This we'll discuss first with the sickle cell anemia. This sickle cell anemia, it is an autosomal hereditary disorder. It is an autosomal hereditary disorder, And this sickle cell anemia, it is because of the caused by the point mutation of a gene, caused by the point mutation of a gene, and it results in the distorted RBCs, what are called as sickle-shaped RBCs. Sickle-shaped RBCs are formed in sickle cell anemia, or erythrocytes, so it is C-shaped RBCs. And this, how this sickle cell anemia occurs? It occurs because of the substitution of an important amino acid. This in sickle cell anemia, glutamic acid, amino acid glutamic acid is replaced by valine. So in case of normal hemoglobin, we have glutamic acid, and this glutamic acid is replaced by valine in case of abnormal hemoglobin or sickle-shaped RBCs. And this occurs, this is at the sixth position of beta globin chain of hemoglobin. That means in the normal hemoglobin there is glutamic acid, and in this abnormal hemoglobin the glutamic acid is replaced by the valine at the sixth position. Actually this hemoglobin, it has two chains, two alpha chains and two beta chains. Hemoglobin has two chains, two alpha chains and two beta chains, and in the beta chain of the hemoglobin at the sixth position, the glutamic acid is replaced by the amino acid valine. This glutamic acid, this is hydrophilic, and this valine, it is hydrophobic. There is a difference also. This is water loving, this glutamic acid is hydrophilic, and this valine is hydrophobic, it is water hating. So what happens in case of in case of this glutamic acid, what happens? This if we'll take, this is normal hemoglobin, or we can say that it is a normal hemoglobin. It's a normal cell, normal RBC. There is, we'll take genetics of this sickle cell anemia. We have gene, or what we call as DNA. It is GAG. Then its counterpart is we'll discuss the genetics of this at the molecular biology. We'll discuss sickle cell anemia. We have normal RBC, we have got DNA in that is GAG. We have got GAG. This G, this is one strand of the DNA, the other strand is G, always pairs with C, A pairs with T, G pairs with C. This is other strand. These are two strands of the DNA. This is what is gene. Then this is, it will form RNA. RNA is single stranded. So we have C, this is template strand. Template strand. G pairs with C, pairs with G, T pairs with A, and C pairs with G. Then there is what there is glutamic acid. There is glutamic acid at the 6th position. There is glutamic acid at the 6th position. This is normal protein, normal hemoglobin, we can say that. Then in case of what happens in case of sickle cell, what is abnormal hemoglobin? How? Abnormal hemoglobin. Then we have gene, same G, T and G. So there is a point mutation in the gene. So obviously G pairs with C, T pairs with A, and and G pairs with C. This is template strand. Then we have got RNA. This is template strand. Then we have got RNA. This C pairs with G, A pairs with T. But in case of RNA, we have, instead of thymine, we have uracil. So there would be GUG. So there would then translation. Then translation, there would be valine at the 6th position of the globin chain of hemoglobin, of the beta globin chain of hemoglobin. This is what is abnormal hemoglobin. So we have seen that this is the major effect of this point mutation in case of sickle cell anemia. Now what happens, this is the major effect of the point mutation. Now this sickle shaped RBCs, that means C-shaped RBCs, they cause the secondary effects. For example, we have normal RBCs. This is normal RBC, and this is sickle shaped RBC. Sickle shaped RBC. This sickle shaped RBC, this is normal RBC having normal hemoglobin. It is having normal hemoglobin. And this sickle shaped RBC, it is having abnormal hemoglobin. It's having abnormal hemoglobin. This sickle shaped RBC, it produces secondary effects. This sickle shaped RBC, it blocks the narrow capillaries. As a result of that, because of the distortion of this, because of the distortion of the shape, it blocks the narrow capillaries, and the blood circulation that gets impaired, that gets disturbed. As a result of that, the vital organs like brain, lungs, they do not receive the proper blood, and which are the secondary effects of this sickle shaped RBCs. So this sickle cell anemia, it is caused by abnormal hemoglobin. We have normal hemoglobin. We have normal hemoglobin HbA and abnormal hemoglobin HbS. This is normal hemoglobin and present in normal RBCs. This is sickle shaped hemoglobin present in sickle shaped RBCs. If we have homozygous individuals, HbA, these are normal individuals, we have HBS. These are sickle shaped. These are abnormal individuals with abnormal hemoglobin. Or we can say that sickle shaped individuals. Sickle shaped individuals with sickle shaped RBCs. These are individuals having sickle shaped RBCs. Whereas these are homozygous individuals. Now we have got heterozygous individuals or heterozygous individuals are heterozygotes. We have HBA and HBS. These are simply called as carriers of sickle cell anemia. These are carriers of sickle cell anemia. These carriers, these heterozygotes, in them the RBCs are normal. They have got normal RBCs, whereas these sickle shaped individuals with sickle shaped RBCs, these individuals die in the childhood because of the anemia. Actually, what happens in case of when there is deficiency of the oxygen at high altitude or during exercise, these sickle shaped RBCs, in case of this, where in these sickle shaped RBCs, they have got what? They have got abnormal hemoglobin. This abnormal hemoglobin, it is having a hydrophobic valine present at the 6th position that forms complementary bonds with another hemoglobin molecules which are present in the RBC. As a result of that, this distorts the shape of the RBCs, and the RBCs become sickle shaped, or they become C-shaped, which results in the disturbance of the blood supply to the vital organs. And these are the secondary effects of these sickle shaped RBCs. So this is the normal major effect, is that the glutamic acid is replaced by valine, and the secondary effects are these result in the damage to the vital organs because of the poor blood supply. Then we have got to this. If we will marry two heterozygotes, what will be the result? If we marry two heterozygotes, we have HBA, HBS, and these are heterozygous individuals, are carriers for sickle cell allele. Then HBA, HBS. Then these are, we'll get children with three phenotypes. We'll have this one, HBA, HBA. This is one type, HBA, HBS. This is another type, and HBS, HBS. This is another type. This is in the ratio of 1:2:1. And these are simply, this is normal individual. And these are carriers. These are heterozygous individuals, carriers of sickle cell allele. And these are homozygous abnormal. That means they are having abnormal hemoglobin, and these die in the childhood because of the anemia, even under normal oxygen amount. These individuals die in the early age. But the allele for the sickle cell anemia, that is still found in certain populations, especially it has got survival value, and it is found in malaria infested areas. It has been found that these heterozygous individuals, the gene for the sickle cell anemia, it is still found in the population because it has got survival values, especially in malaria infested areas, for example tropical Africa. It has been found that this plasmodium, it cannot penetrate the RBCs. This plasmodium, it cannot penetrate the RBCs of heterozygous individuals, HbA, HbS. As a result of that, these individuals do not get infected with the plasmodium, and they do not get the disease. So this sickle cell allele for sickle cell anemia, it has got importance in case of malaria infested areas. This was regarding the pleiotropy, and what are the major effect is substitution of amino acid, glutamic acid is replaced by valine, and then there are another effects of this sickle shaped RBCs. Then we'll discuss phenylketonuria. This phenylketonuria, it is also a point mutation. What happens in case of phenylketonuria? There is the building up of phenylalanine in the blood. Amino acid phenylalanine builds up in the blood. Because of the mutation in one of the genes which is responsible for the synthesis of the enzyme, and that enzyme is responsible for the processing of the phenylalanine. As a result of that, when that enzyme is not formed, obviously this phenylalanine gets piled up in the blood of the individual. So it is a gene, and it synthesizes an enzyme that is called as phenylalanine hydroxylase. Phenylalanine hydroxylase. And this phenylalanine is responsible for the processing of phenylalanine amino acid. This is the major effect of this point mutation. Now, what are the secondary effects? Those persons which are suffering from this phenylketonuria, where this enzyme is not synthesized, they have severe secondary effects. Like they have eczema, that means skin problems. They have mental retardation, excessive sweating, these are the other secondary effects of this. These are the secondary effects of this and pigmented patches, eczema, mental retardation, excessive sweating, and pigmented patches on the skin. These are the pigmented patches on skin, and these are the secondary effects of this point mutation in case of phenylalanine. So in case of ketunuria, so we can say that pleiotropy is simply the multiple effects of a gene. When one gene influences several phenotypic traits together, that is called as pleiotropy, and it can have major effect as well as minor effect, and we have seen in case of sickle cell anemia and phenylketonuria. Thanks.